nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in developing CRISPR technologies, and particularly today I'm going to talk about pooled screening. It's nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in developing CRISPR technologies, and particularly today I'm going to talk about pooled screening. It's nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in developing CRISPR technologies, and particularly today I'm going to talk about pooled screening. It's nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in sort of developing CRISPR technologies, and particularly today I'm going to talk about pooled screening.
So good evening and welcome to the BioGENext series. It's a great pleasure to see so many of you today here at Sikorsky Kyiv Polytechnic Institute at the opening of our second season of BioGENext series.
And by tradition, before we dive into the world of single-cell omics, let us introduce ourselves and say a few words about the BioGENext series. My co-host today is Alexander Hooper, the head of Biotech R&D YP Biotech, the Biotherapeutics Research Unit of pharmaceutical company Uriapharm.
Yeah. And I'm more than happy to introduce Dr. Oksana Mankowska, a dedicated researcher at the Department of Molecular Oncogenetics at Institute of Molecular Biology and Genetics of National Academy of Sciences of Ukraine.
We highly value the opportunity to hold this host and to host you at this beautiful place. So first and foremost, we truly want to express our deepest gratitude to the armed forces of Ukraine, to all the defenders of Ukraine who bravely stands for Ukraine during these 3.5 years of Russian war against Ukraine.
So now, may I ask all of you to stand up and observe a moment of silence to honor the memory of all our defenders. Thank you all. Yeah, and let's see our slides. Just a few clicks. Yeah. So just a few words to remind you of what the BioGENext is. It's an initiative created by scientists and for scientists.
About 1.5 years ago, our teams from Uriapharm R&D and the Institute of Molecular Biology and Genetics, National Academy of Sciences of Ukraine, decided it was the time to combine our efforts towards a very important goal, to build a strong biomedical research community in Ukraine and create a platform for academia and industry to meet each other, learn from each other, to perform groundbreaking research.
And the vision of such platform materialized into the BioGENext initiative. Exactly a year ago, actually in the same dates as today, we started by hosting our very first BioGENext conference here in Kyiv. It became a starting point, providing not just the knowledge, networking, and partnership to all the participants, but also the inspiration and new perspectives on their own research.
But this was only the beginning. The conference has proven that there is great demand for an environment that allows live discussions of various methodologies, practical aspects of their implementation, and gaining feedback from experts in the field with extensive hands-on experience.
So we launched a BioGENext series, a spin-off of educational initiative jointly created through collaboration between Uria Pharma R&D, Institute of Molecular Biology and Genetics, NGO, Unia Scientific, and YP Biotech, to help you master and increase your proficiency in cutting-edge technologies and methodologies and learn from the best experts in the field.
Thanks to our academic partners from leading universities who actively supported the initiative, the two of the first 3 lectures in the series we held at Taras Shevchenko Kyiv National University and Kyiv Aviation Institute, alma mater for many of you who are present here today.
Yeah, and the lecture focused on the selected state-of-the-art and emerging methodologies ranging from structural biology to multi-omics and organoids application, all of which typically require input from the highly trained experts for correct and effective implementation.
We are truly happy that you have joined us in this wonderful journey. Now it's time to launch the 2nd season, and we are truly grateful to Sikorsky Kyiv Polytechnic Institute and the Faculty of Biotechnology and Biotechnics for joining the initiative and kindly hosting today's event.
It's not a coincidence that we are talking about the biomedical research and development here at Kyiv Polytechnic Institute, as this university has an extensive experience of more than 25 years to upbring new generations of biotechnology specialists. And now I would like to invite Tatiana Totoseychuk, the Dean of the Faculty of Biotechnology and Biotechnics, for a welcome vote. So please welcome Tatiana.
Dear friends, dear colleagues, I'm very glad to welcome you to Igor Sikorsky Kyiv Polytechnical Institutes at these lectures, where our faculty, the Faculty of Biotechnology and Biotechnics, is a part of this interesting project that started by BioGENext series. This academic year, our faculty turns 25. And today's event is taking place as part of our anniversary celebration. At these lectures, at this event, our faculty connects with the scientific project BioGENext series, not only this exactly place, and common interest in dissemination current knowledge in biotechnology and biomedicine, but also the guest lecturer, Maryna Korshevniuk, who is a graduate of our faculty. Some years ago. And because of this, we are very glad that our graduates can already share their knowledge and research results with the colleagues and young scientists, as well as show the students and all the who interested in biotechnology the direction and prospects of their future activity. And I have no doubt that today's lecture will be interesting both for scientists in biomedicine and biotechnology and for those who are just starting and just beginning their journey into the secrets of this innovating scientists and practical area. So I wish all participants interest in scientific communication, gaining new knowledge for young people, and in general, discovering new opportunity in biotechnology and biomedicine. Thank you.
Thank you, Tatiana Serhievna, for such an inspiring words. And just one more organizational issue, that in case of a raid, we will still here and proceed with the event because the class space is designed as a shelter.
Yeah, another organizational note, as our audience is international, we keep English as our formal language for the BioGENext series. However, for our on-site participants, if you, during the Q&A session, feel comfortable to ask questions in Ukrainian, Maryna will be very pleasant to answer to your questions, so do not hesitate, ask more questions, because that's why we all here.
And now let's dive straight into the fourth lecture. It's my great pleasure to introduce today's speaker, Maryna Korshevniuk, PhD candidate in single-cell multiomics and personalized medicine, UMCG, Netherlands. Maryna is also CEO and founder of Apixmed, personalized medicine solution provider. And last but not least, she is a graduate from the Faculty of Biotechnology and Biotechnics of Kyiv Polytechnic University. Maryna, welcome back to the halls of your alma mater, and this now floor is yours.
Super. Hi everyone, and I'm so excited, so I cannot express how much I'm excited about being here and sharing the kind of a little bit of the highlights and knowledge that I earned during last few years, during the last 10 years. So first of all, before jumping to the lecture, I would like to express my absolute gratitude for organizers who trusted me to host me today, but also for you to expressing, and as well as online audience, for expressing the pure interest into the advanced discoveries and innovations in the field of biomedical technologies. So before jumping to the lecture, I would like to know how many of you have been working with single cell before. Just raise your hand. So, okay. Okay, a little bit of proportion. I already see many familiar faces in terms that we were delivering a lot of educational courses, materials, and so on and so forth to democratize access to the bioinformatics education within the biological summer schools, genomics UA initiatives, and so on and so forth. So I hope that for those who are unfamiliar with single cell, as well as for those who are familiar with single cells but unfamiliar with single cell genomics technologies, my lecture will be useful. So with this lecture, I would like to break all the rules in terms that we do not follow the standard schema about like putting the slides as they go. I would like to encourage you to actively be involved, and it's your first responsibility as my audience to be involved in the lecture and think actively about the research question that you might face yourself in these studies. So let's jump to the lecture. The lecture of today is how can we use single-cell technologies to understand better the disease mechanism. As I was introduced by organizers, I would give you a little bit broader view of who am I, what I've been doing during my last 10 years of life, or a little bit less, and again, how I ended up here. So with single cell, I was familiar from 2018 from Max Delbrück Centrum in Rajewski's lab, where I've been using single cell technologies to understand better human brain organoids changes. So stimulating these in vitro systems and applying in vitro single cell technology, single cell RNA-seq to better understand the difference in transcriptomics profile. Further, with a passion to single cell and human brain organoids, I continued in the University of Luxembourg, as well as a scientific expert. And the moment X in my life happened when I started my PhD in University Medical Centrum Groningen in the Netherlands. Thank you. Where I'm working with single-cell multiome to better understand human genetics. Beside my academic track, I have an extensive activities and initiatives Outside Science, first of all, is an amazing company that I founded with my colleagues, where we democratize access to the data analysis from genetics and the other omics modalities for real-world insights and application for the field of personalized medicine. But also, I am an educator. I possess myself as a lecturer for the Genomics UA and other initiatives, as well as founding the organization for mostly medical education and professionals to elucidate the mechanism and insights from personalized medicine. So before I started with intro, I would like to double check, can you hear me well? Do you want me to slow down or everything is clear? It's good. Super. Okay, great. Then we can start with intro. And as I promised, it won't be boring, at least the intro, okay? So with the intro, we will start with like three main principles, like the three main, let's consider these the elephants that are staying on the turtle, or like the three whales that are holding the earth. So I see these three elephants of my talk that you need to keep in mind as a concept. So elephant number one is something that you're absolutely familiar, is the central dogma of molecular biology, first introduced in 1957 by Francis Crick. That's something that every molecular biologist, every biology graduate needs to know. But what actually happens is that during last advances, the central dogma was extended, massively extended with the advance of other omics modalities. And even though the transmission and transit of genetic information hasn't changed that much, we still know that the direction where the information from DNA to RNA and protein is transmitted is not that straightforward and like one-to-one interaction. Therefore, what we see here is that, let me test the clicker, okay, super. What we see here, that from the genetics and genome perspective to the phenotype, the end result of how our biological system might look, there are several modalities that are involved in the signal transmission and the genetic signal realization, meaning that it is very important to fully understand the role of this genetic signal and deconvolute the methods, like the ways the signal is transmitted to the final phenotype. And for this, we need many advanced technologies. So, elephant number two is that we also know that genetics is not only fundamental science that we all learn in high school, or many of you learn within the university curriculum, but it's also something applicable to our everyday life. As such, we know that the genetics has a strong link and the basis of the occurrence and predisposition to many diseases and conditions. Therefore, we also need to understand how this kind of transition from a single genetic signal to the final complex phenotype is happening. So one of the common way, and that what you totally have first thing in mind when you think about genetics, is monogenic diseases. We know that the one single change can affect the whole phenotype by breaking the links and systems in the, on the tissue and cell level. But the case of monogenic diseases is very rare. So fortunately, a small proportion of our population is affected by those, and this still remains in the scope of the medical genomics, and it won't be in the scope of my lecture. However, something what is much more applicable to our everyday life is common diseases. Common diseases studied often by genome-wide association studies, where you know that several predispositions to conditions are affected or associated with the genomic variation, genetic level variation. As such, my talk will be allocated to understand how these complex diseases, where not a single variant but many variants are affecting the phenotype, are realizing within different omics modalities. So this is kind of the contrast that I would like you to just have a glance at. In case of monogenic diseases, we know that the one signal is kind of linearly transmitted to these modalities, affecting a very small proportion, or a very small number of the entities which are affected, kind of impacted by this single genetic signal. However, in contrast, in the complex or polygenic diseases, we know that there are thousands of the signals that can affect or can kind of guide this predisposition to the diseases, affecting many layers, from putting the light from the chromatin perspective, affecting mRNA interaction and transcriptomics, proteome as the modality the closest to phenotype, and the final phenotype in the end. And in terms of the complex diseases, they affect up to 80% of the world population, might affect each of us at some point of our life. That is why, to fully understand this mechanism and be able to prevent them in the proper time, in the proper target, we need to fully understand those mechanisms. So that's why we come to the last elephant from our intro, which is the fact that genetic variants, which can be linked to these diseases, They also do not work steadily. They do not work in a single context, kind of the same in every context. The single genetic variant has different roles if it's the variant operating in brain, in liver, in blood, and so on and so forth, meaning that these genetic variants have a very strong context-specific manner of action. So this is kind of the elaboration that even though we know that there is a huge proportion of genetic variants associated with the disease, we don't know this underlying principle. And the underlying principles are often very context-specific. Again, polygenic risk score, but what is important that having these three elephants in mind, like knowing that the central dogma was significantly extended with the advance of high-throughput technologies, knowing the fact that the genetic is affecting the disease predisposition and states, and knowing that all of this action and interplay from genetic level to the final omics modalities is happening in a context-specific manner, we can, for today's lecture, we can ask ourselves the question, how can we grasp the results of this context-specific action, and like, how can we use the advanced technologies and grasp this context-specific action of genetic variants to better understand and prevent in the future diseases? So, with this, I would suggest the following structure of the lecture. We will have three main parts. They are disbalanced, so if you are tired of the part number 1, no worries, part number 2 and 3 are shorter. But the idea is that we will first cover the landscape of single-cell technologies and understand how talkative are our cells, how much power of information can a single cell provide to us. And number 2 is that we will understand how blueprint on action is operated. Blueprint is our DNA, meaning that how, in what context the usage of DNA can happen. And last, I will kind of give you the food for thoughts for future, such as like where the field is moving, what you as a young scientist or experienced scientist, what you can consider for your future research, and what is kind of the whole dynamics of the field, which might be or might not be relevant for you. But the goal is to make it relevant, to make it kind of applicable, and educate you about the landscape of the contemporary technologies. So long story short, I put myself as several goals. First of all, I would like to cover the basics of the single-cell technologies, put the light from the genomic perspective, part number 2, and also have a vivid and a fruitful discussion afterwards about the future perspectives about single-cell technologies. So let's start with part number 1 then. What we know and what many scientists are familiar with in the wet lab. Usually when we are operating with a transcriptome or genome, we do the following, or like, okay, 15 years ago, like 10 years ago, I was doing the following. I was coming to my lab, getting the sample, whatever, it's like a mammal, plant tissue, or microbiome, whatever, it doesn't matter. So I took my sample, within a research question in my mind, I was homogenizing sample and conducting the further analysis, proteomics, transcriptomics, genomics, whatsoever. And the idea is that I was interested in knowing the expression profile on genes in terms of the transcriptomics, the protein abundance, or like genomic sequencing. But the problem of this type of analysis, even though it's super powerful still in this moment, the goal is not to kind of lose your passion or demotivate you that bulk technologies are not relevant at all. But even though this analysis is very powerful, it still doesn't have the full recapitulation of the heterogeneity within system. How many of you have been working with microscopy images? Super, so more than single cell. Okay, then it's like analogy is more familiar for you. Ideally, it looks the following, that you've probably observed within the microscope image that the cells look differently. Even the cells within same cell type are not the same at all. Why then we think that they are the same on expression level? Obviously, they are not. And our goal, the scientific community, is to find the instruments that are representing this and kind of providing the ability to explain this heterogeneity on the level of biological samples. So bulk methods, even though they are significantly powered by next generation sequencing, and then they are allowing this research at scale, they still had some limitations. As such, we were not able to understand the inter-sample heterogeneity of our research system, like biological system. With the advance of microfluidics technologies, with the advance of the fact that now you can treat a single cell as a single sample, it became possible to tackle this heterogeneity and understand the differences within the cell profile, or even the differences on the level of a single cell. That is why it kind of was attracted, like, as a whole, the scientific community, it was recognized by the method of the year from as a single-cell RNA-seq, the single-cell attack-seq, sorry, not attack, but multiome, and the single-cell spatial technologies. It was widely recognized as the method of the year, and that is why it is still a very hot topic to jump in if you are considering the directions to start with your bioinformatics studies. So this kind of things is learned that next generation sequencing together with microfluidics forced significantly the appearance and like democratization of the single cell. For anyone who have been attending single cell course, you've seen this slide, you are tired of that, I know, but the idea is that for those who are unfamiliar, that the throughput and like the scale of the single cell technologies, sorry, I have some problems. So that like starting from a very few number of cells from 2010s and so on and so forth, by scaling these technologies, the throughput of this analysis is massive at this moment, like coming up to 5 million of cells with the contemporary droplet-seq-based methods. And the idea is that this field is still very rapidly developing and the scale is growing. Basically, here we see that no matter what kind of the cell capturing approach you use, no matter how you collect and isolate your cells, it's still important to tackle the proper methodology to be representative enough for your research question, but also to reproduce you the enough cell capture, like enough cells in the end. What is it dependent on? This graph is complex, I know, I don't want you to remember it, but I would like you to look at a particular point, such as there are several protocols. One of those, like this drop-seq base, is commercially available, that are more often used in a field than others. And the idea is that, and this is important thing, that single-cell technologies are not the same. Like single-cell technologies are very much different in terms of protocol, in terms of modality you capture, in terms of your research question and ability to be representative and informative enough for that. So in a single cell, you can differently approach the cell isolation and clustering procedure. You can use the FACS sorting, like fluorescent activated cell sorting, together with microfluidics. You can use the droplet-based technologies together with microfluidics. You can use nanovalves, such as like huge wet lab plates for like massive amount of cells to isolate your cells and then treat the cells as an individual volume. But the idea is that you still need to understand that you need to isolate your cells. You need to be able to track your cells in the final data set when you will be doing the data analysis. So the technologies are the following. The drop-seq based technologies, such as 10x technologies, is one of the most widely used. It is the 3' end sequencing technologies, and then you can give the relatively sufficient number of the cells per assay. This is one of the reasons why this technology is very easily adoptable from the commercial perspective, and this is something that you might be working with if you will start your journey with single cell. In contrast, there are also other technologies which are not necessarily tackling the mRNA or mRNA, yes. Okay, sorry, I hope my mic is dead. Okay, so basically from the SmartSeq2, SmartSeq3 perspective, you can do the full length coverage and then find not only the poly-A tail RNA in your dataset, but this is like step by step. So basically, the number one and core principle of single cell that I try to phrase verbally, and this is visualized here as it is that you work with the single cells which are kind of isolated in a single volume, then you creating the emulsion to isolate the volume of a single cell within, like from the other cells, but also it is important that within this volume, within this small, it's very tricky though, within this small kind of granular of the emulsion bit itself, you can also put something to track your signal. So how it works, you have like a microfluidic system where from one stream your cell in the hydrophilic volume comes, and from another it's like the other type of substance, but with this particular metal bit visualized here. Jesus, it's really very difficult. Okay, trust me, visualized here, visualized here from green and blue. Basically, the idea is that you combine yourself with something that is representative for this cell type specific signal. This something is able to capture your mRNA because, from the following reason, because it has like on these black sides, it has the poly-T tail, which is complementary binding to poly-A tail from your mRNA. And further, having this information in mind, you can trace your cells back from the UMI and cell barcode, meaning that cell barcode is some kind of oligonucleotide sequencing specific for this bit, like for all of these kind of oligonucleotides with this hairy ball, and unique molecular identifier is specific for this molecule itself. So basically, what is this like green and blue bubbles or circles is a small metallic particle which is ligated to different oligonucleotides which are further allowing you to track the signal from your single cells. So if you would like to know more, there are many interesting visualizations, like useful visualization in 10x genomic sites, which can be also like used for further study. Out of scope, so let's continue further. Considering the information that we've been working with poly A tail, we also have to be aware about the disadvantages of the final data set that we decided to go for. First of all, if the poly A tail of your molecule is not long enough, it will not that sufficiently and effectively end up in your library. Also, as we work with poly A tail, it's like preprocessed mRNA, meaning that you will not see that many unspliced transcripts and so on and so forth. This method is also sensitive and biased for the weakly expressed genes, and you will not see that many weakly expressed genes, which are extremely important for the regulation perspective, but still this method will not allow you to do this because like it's a power of many. You will have many more ribosomal genes than, for example, genes of transcription factors. And so on and so forth, like complex genes having the complex structure of or a big length, as well as GC content, as it's kind of implicated by amplification step, or the degraded transcript, something obvious because it's not poly tail, that you will not see it. So the take-home message is that in a commercial system, as 10x Genomics DropSec or something else targeting the poly-tailed mRNAs, you will face several disadvantages that you need to be aware of in advance. So in case you are planning the experiment by learning non-coding RNA, 10x technologies is not something for you. Consider other technologies, full-length sequencing like SmartSeq2, SmartSeq3, that are more representative for your research question. Okay, so what's going on further? After having your cells preprocessed and the library, you can further go into the amazing types of analysis. So steps up to here, yes. So steps up to here are pretty much standardized. They're still specific up to your sample type, meaning that if you work with blood, if you work with brain, you will have different QC metrics, but you will follow the same pipeline. So starting with sequencing reads, you generate the count matrix. This is something probably further on, but like provided by ScanPy or CRAT packages. So as soon as like you Google single cell data processing, you have like everything you need. That's like these two main packages. And you further continue with until the step, like after QC and the integrations, where you can cluster your cells together. This all is based on the principle that the cells within one cell type have similar expression profile, meaning that they will be clustered together and that are further can be considered as a one cell sample type. Count matrix, yes. So basically with the cell annotation, when I've been starting, the cell annotation was the following. You play around with different clustering thresholds and the hierarchy of these cell types to fully understand that if the cells you're looking at, they are representing one cluster. But now the world has moved further, fortunately, that now you treat your data set to annotate your different cells using the external reference. It means that if you are working with blood, I'm working with blood, for example, in my research, you are not the first one who works with blood. Many other people been working with this data, prepared, like, again, from these commercial companies, prepared high-quality data sets so you can easier work with your blood, like, in terms of sample size, sample type. So basically, then you can use somebody else's data, which can be better in terms of quality, better in terms of sample size, better in terms of transcript coverage, and so on and so forth, to further annotate your cells. Basically, this is kind of called the reference-based mapping. Having your data, you even do not necessarily have the need to annotate the cells yourself. You can use external transcript, again, provided within this reference clustering tutorial. So, and then the kind of, if first part of preprocessing is boring, this part is exciting. So having this cell type information, what can you do? You can do really a lot of interesting stuff, which are purely dependent only on your research question. I will start with this kind of clockwise. like this cell clustering, cell type identification, even though that we just say that it's a part of this kind of pre-processing, it is still a very important point if you work with, for example, rare cell populations. If you are kind of like checking how much one sample from cancer treatment is responding in comparison to other, there can be populations of cells that appear only in this kind of context-specific manner. So identifying this complex and rare population is the key. Again, if you are working with while comparing two different conditions, you can, like from healthy and disease sample, you can compare how much one of the cell type is more abundant rather than another cell type. This is kind of the cell composition analysis. And then if it's like statistically significant enough, you can state that, okay, so with this disease state, we see this kind of whatever, the splash of the activated CD4 T cells and so on and so forth. So idea is that it's still important to look into the cell composition and like the coverage of the representation of the single cell type after like within two conditions, or within like even you have one condition between male and female, between old and young, and so on and so forth, like pure space of creativity there. Again, if we talk about this comparing health and disease, the differential gene expression. Similarly, pathway gene and scent enrichment analysis. If you've been working with bioinformatics, these terms are very familiar for you because this is something that comes from bulk. It's not reinvented for single cell purposes, but still. But something that you can do only with single cell data is that pseudotime and lineage interference, meaning that you can trace within your data set how the progenitor cell was kind of generating and like affecting this hierarchy in terms of differentiation perspective of your data set. Like, let's imagine hematopoiesis. We know that it comes from the stem cells and then the hematopoietic stem cells and then differentiated through lymphoid and myeloid lineage. Like, knowing exactly with these breakpoints and like subcell types for this differentiation, you can already see from your single cell by using like pseudotime and like different velocity metrics. Similarly to the cell state, knowing if your cell type in the whatever in the mitosis sphere, if like the cell state is like activated cell state, or like if something is happening to your cell, by having information about specific genetic markers that are expressed and allocated to a particular like cell cycle type, or like the cell type itself, like cell state itself, it is informative enough to make a conclusion about this transition between cell types. Last two are my favorite because it's my passion. So first is ligand and receptor analysis. Knowing the genetic profile, or like transcriptome profile of your cell, allows you to approximate the ligand abundance and receptor abundance within the cell within the same and different cell types. Meaning that without having information about the proteins on the surface of this phase, we can still suppose or kind of assume that there are some kind of ligand and receptor analysis interaction happening, and that's the way we understand how different cells within the same cell type or different cell types communicate. I find it super exciting. I would love to learn the genetics basis of this, and maybe it will be my next project, but we'll see. And cherry on the top is, of course, network modeling. We know that the genes do not work in an isolated manner. We know that genes are co-expressed, they are down-regulated, up-regulated. One genes regulate other, another to suppress other, and so on and so forth. So knowing the network configuration within your cell type is super crucial, it's very important, and you need to be equipped with the tools, and you need to have a high-quality data set, because crap in, crap out. Like, this is kind of the thing. With this kind of set of technologies, many advances were made. The biggest is, of course, human cell atlas, millions of cells allocated in a single the same site. So if you are looking for inspiration to start with a single cell data set, check out this human cell atlas. But also other data sets, like huge consortia, like Tabula sapiens and Tabula muris, where all the organs, most of the cell types of human and mice body correspondingly were covered. So even it's like some questions about sample size, not me to say, but still you can now see what are, like for the same donor, what are the expression dependencies for the different, cell types and different organs covered. I find it super exciting, and I think these initiatives are yet to come. So yeah, it's very interesting to know what is kind of the current state and what are the superstars of the in terms of project in the single cell field. And now to wake you up, I would like to ask you, how do you think are the bulk technologies dead? And why not? I didn't know what to ask, but yeah, it's like... Okay, why not? Okay, because I already said like bulk is not dead, so, but why? Why bulk is not dead? What's your gut feeling? It's like... For what purpose? Sample space. Okay, interesting hypothesis. I will repeat that sometimes bulk and single cell are used together, because all my audience haven't seen, because it's like to increase the sample space in your theory. Yeah. Yeah. Okay. Where to look at? Yeah. Okay. DNA is a baseline for organism. We do not study DNA that much on single cell level. You're right, it's elephant number one, but it's more about RNA. Let me elaborate then. So indeed, bulk technologies can be used for complementary research or research like hypothesis validation. So bulk technologies are there for tens of years. They are not invented at the same time of single cell. So if you design your experiment, like keep the benefit of balancing single cell and bulk, where you can also like tackle the high sensitivity is something you can tackle, but also like this resolution of single cell, but also the high signal from bulk, because in a single cell you do not have the coverage of all the genes. It's kind of the very stochastic process where you end up with a data set where like 95% are zeros, and then you can kind of do some magic to find the signal itself. It doesn't work that, it does work, but it can work better. But in bulk, you have a baseline. You always have a signal for a gene if it's present, like lower and higher, like after normalization, you can state this. So meaning you can use this for hypothesis validation or for complementary study, like together with a single cell. But also what you can do is that you can like take the benefit of the huge amount of bulk data set, bulk RNA-seq data set generated before. The methods which you can use is called deconvolution. Like here is like not a call to action to check, but for you to understand that even not without having the luxury to do the single cell analysis, you can find the relevant or like generate yourself relevant bulk data set and then apply the computational technology called deconvolution, where by estimating the expression profiles of a bulk sample, you can assume the cell type proportion and the corresponding the profile of this of the cells. This is kind of another kind of positive moment that, like with the single cell data sets, which can sort millions of euros, you can use the cheaper bulk, which can be a good hint and like good complementation for your hypothesis there. Okay, we are on the middle of the part number one. So we talked mostly at this point about RNA-seq. However, obvious answer on this question, whereas one modality, is it enough? Is it not enough? Is it representative? Good? Not? Is like, what are we missing? The obvious question is like, one modality is representative enough in a very small amount of cases, meaning that as more modalities we have, like coming back from this example where the genetic signal is transmitted through multiple omics modalities, like as many more modalities we know, as better we can recapitulate or like design in silico or like whatever, better understand our system. With this, there is like a huge amount of single cell multi-omics technologies are used, and the idea is like, yeah, I see many of you getting photos of presentation, write me on LinkedIn, I will share it for free, so like don't distract yourself, focus on content. So basically, you can cover several modalities within the same cell. Here is the kind of this type of coverage happening. Let's eat this huge elephant piece by piece, where, for example, we can study the gene expression together with the DNA modification. This is kind of GT-seq, like the genome and transcriptome sequencing. And this kind of analysis is very applicable for somatic mutations, where we would like to track the changes on the level of DNA together with the consequent changes on the RNA. Nevertheless, not only genome can be covered with the single-cell multiome technologies. There's something that is much more widely applied, at least in my field of studying blood of healthy people, is the understanding the epigenomic regulation. The widely applied and applicable protocol where you can study both RNA expression and the open chromatin, like the regions of chromatin which is not dense, which is kind of active for whatever action is required, like regulation, silencing, and so on and so forth. So basically, you can study together RNA and the open chromatin, which is kind of the method called from ATAC-seq. Together with this, following with the histone modification, methylation, you can study this together with RNA and the level of this type of epigenomic regulation. This is extremely important because then you can understand not only what's happening, but also why. Like if you know what is kind of the trigger on the methylation or open chromatin level, you also know like, okay, maybe that's kind of the hint. Maybe this region is more relevant for me to zoom in. Like maybe this region I have to, whatever, engineer in lab to test these primers, transcripts, and so on and so forth, to design the system that we will talk about in the part number 3. And lastly, especially for those who work with immunology, immunohistochemistry, is studying the surface protein of the cells. Together with the RNA, you can take the advantage of studying the surface protein, and if you can see why it's happening, like why the hell this is no DNA, this is kind of attack, this is all of kind of nuclear sequencing, nucleic acids, how come these epitopes come here? The trick is that you need to know what to tackle, what kind of epitopes, and also, you know, you need to modify your antibodies so that you can further use and find them back in your library. So this is very important to understand, like how much the transcription abundance is linked to the protein abundance of the target molecules. And again, with RNA and DNA-based, like DNA-focused research, you can do like in an unbiased manner. So you can check whatever it comes into your library. With epitope and the similar technologies, you need to know in advance what to look at, because like there is no, if you don't have specific antibodies, you will not have enough signal. So now the question for you to keep in attention, what modalities are missing in this slide? Any guesses? Raise your hand if you have an idea. Only molecular biologists are here. Okay, please. Say it again.
The proteome, maybe?
Proteome is kind of barely covered by site-seq, like the epitope. But you're closer. You're getting there. So metabolites, exactly, because it's very difficult to tackle those. Like metabolites is something that often difficult to be tackled with antibodies, and then like the scale of experiments has to be huge. But also from the further analysis perspective, it's not often that used. And again, you were right partially about proteome, because proteome is like epitope is something easily to be recognized, but the let's call it proteome-wide screening is much more tricky. You need another type of methods. Everything what we talked about is NGS-based, and for proteome and the metabolome, you need like mass spec or like HPLC-based system further with mass spec. So basically, I'm not discouraging you to forget about metabolome or so on. I'm stating that by selecting the right single-cell multiome methodology, you need to know what to expect in your dataset. It's super obvious. I'm not educating you here, but the idea is that, like, know the power of your instrument and know the restrictions. So, closing the part number 1, how can we use multimodal dataset, like knowing information about different omics modalities from the same cell, or having the same sample size proceed, for example, with ATAC-seq and RNA-seq, what can we learn from that? First of all, we can integrate this data, and this data integration means that we can have the complete picture about the modalities activities from the same cell, or like the cell type, if like this comes from 2 different samples. This is, again, another level of information and deeper our knowledge about what's going on there. What is also interesting, especially in the context of machine learning technologies and so on and so forth, is that by knowing the behavior of one modality, dependency of another modality in one design system, in one data set, you can you can assume or you can predict what's going on in other data set where you have only one modality. For example, we know that like, if gene is expressed, the chromatin around this gene is open, meaning that as it is more likely that for expressed genes, the chromatin regions will be open, and vice versa. So if the chromatin is open, genes are likely to be expressed. So you can impute and kind of predict the unobserved modality and somehow use it like with asterisk, but still for your research. And lastly, following this idea about the open chromatin and the transcriptome, you can also augment this or pre-filter the data you work with. As such, it's a whole pain of everybody who tried with single cell. As such, you have a hell amount of zeros in your data set. Like the zero and data sparsity is the real issue. It's an alarm, right? Okay. So data sparsity and zeros is a real issue, and then by having this information from different modalities, you can kind of minimize the number of these zeros and at least have like the probability that this is not a zero or like it is something. So something very useful to improve your signal, increase your sensitivity, and better and wider your knowledge about the particular biological system. So with the downstream analysis closing the part number 1, we can like having more modalities, we can better do the network modeling because now it's like not inside one network with itself, but with the several nodes connected. You can build these links by correlation, association analysis, and so on, and have the dependencies of how, for example, chromatin openness is affecting gene expression or DNA variant is affecting those two. We will talk about that. Also, we can improve our clustering and cell type identification because we increase and enhance the signal. Now we are more kind of confident about these cells. Like these cells are high quality cells. We know that these are like lower zero number and so on and so forth. And we can now better detect rare cell type or annotate cells. And lastly, you can do not only differential gene expression, but differential something expression, differential open chromatin, differential methylation, and so on and so forth. So your kind of ability for comparison, like the relatively simpler test, is much more wider, and meaning that your insights can be better applied and extrapolated for biological knowledge. So let's make a recap that, first of all, single cell is super powerful, one love, like recommend for everyone single cell, and it allows to study different cell type population. You can work with single cell not only with RNA, but also with our other modalities. And considering that single cell RNA-seq is a luxury, multiomics is a luxury in a power of 3, but still you can work from the data perspective without the need to generate this data. And also, working and looking into several modalities is boring, so you need to understand how to better integrate the signals and create the better representation of your biological system to find a more proper answer from your research question. So, as I promised, part number 2 and 3 are shorter, and with part number 2, we will talk mostly about how genetic works, and this part is called, that's the reason why it's called Blueprint in Action. Idea is that we stated that for polygenic diseases, there are many variants contributing to the common phenotypes. The way this transition is happening is obscure. We don't know, like, what's happening in the Jiva studies is the following. You know that there is like the 2 different categories of people, like let's call it with the categorical like diseased and healthy, and there is also like the set of the variants which are likely to appear more often in the disease group or likely appear more often in the healthy group. So you have only associations, but correlation doesn't mean a causation. You don't know like if this genetic variant is contributing in general, or this genetic variance is mimicked by other variants, or it's like co-inherited, or whatever action is happening, or like maybe it's not this genetic variant is relevant, but like the open chromatin around it, like nobody knows. It's still relevant, but from the other mechanism. So long story short, we don't know the mechanism how the transition of genomic signal is affecting the end phenotype. And so far, zillions of not zillions, thousands of associations from Jiva's catalog were made, and we work a lot with Jiva's catalog. It's a little bit of pain, but we know that more than 500,000 of associations are made for 15,000 of curated traits and phenotypes. But again, we don't know why. Like, what are the mechanisms? There are some hints. People from medical genetics like to follow this mechanistic approach, like MTFHR or FTO are affecting that and that, but reality is much more different and difficult, that you need to follow a network approach, you need to study the mechanism within the context, like the action of this genetic variants in the relevant context. So basically, one of the ways to study this, and like at least to make a hint, is so-called QTL analysis. QTL analysis is the topic of my PhD, so if you would like to know more about QTL, subscribe, but idea, I will make a short brief. So basically what's going on? By having information about the genetic variant and something quantitative trait, like the level of gene expression, like protein abundance, like scope of methylation, and so on and so forth, chromatin openness, you can do the quantification and check the correlation, how much this genotype of your kind of target genetic variants is associated to the phenotype on this molecular layer. Not the phenotype like the type 2 diabetes, but the phenotype like level of RNA set2 expression, for example. So basically with this analysis, by having these quantitative traits, and in this case like expression traits, we can better understand why behind some of the jivas, like what can happen behind them. Like, okay, this genetic variant might affect this expression in this tissue, in this cell type, in this context. And this kind of context, cell type, and this like, in general, mechanism of action is giving the hint maybe how to prevent and like what to tackle and zoom into treatment or prevention of these diseases. Also, what we know that the genes are not operating in a single manner. Genes are often co-expressed, and moreover, this SNP can affect single nucleotide polymorphism, SNP can affect the gene co-expression itself. And here again, surprisingly, we can follow the quantitative trait loci analysis, QTL studies, where in this quantitative trait, it will be means of co-expression of these genes. And again, this is like a very simplistic view, but a single variant is providing the network effect, affecting the co-expression of many genes, and like many genes themselves, like usually a single gene, but like many co-genes. And ideally, what you would like to do the follow, again, it's like about that. It's not only e-genes that can be covered by these eQTLs, but also methylation QTL, the chromatin accessibility, histone modification, like microRNA, non-coding RNA, protein QTL, like tons of these types of analysis can be done. And the idea is that no matter what you do, you would like to know how much what you observe is similar to the jiva signal. How much like what is similar seen from this genetic perspective, from this jiva studies, how much the signal is overlapping from the QTL signal. For this, you use colloq or colloq-like packages, and you try to overlap these genetic signals from jivas, again, here, from jivas and the eQTL perspective. That's why we see the linearity, because we would like probably to prove kind of the causality that there is a dependency between these two. So we overlap and see how much this jiva signal is overlapping, how much it's similar to the eQTL signal. So how it works, you overlap this whatever type 2 diabetes signal and the level of the HLA gene expression. Let's imagine this. And with this type of knowledge, working only with single cell, or like only with bulk even, and QTL analysis, having the jivas information, you can already have the validation of like, are you looking into the right system? Are this variant is actually linked to jivas? Are this variant is in the right context? Because depending on the context, the genetic signal will be different. And then it's kind of the, yeah, it's something interesting to find the most relevant context in the signal. So using this information, you can follow and get closer to causality of the genetic variant affecting eQTL or like effective quantitative trait loss, but also being associated to jiva trait. So recap, in intermediate recap from part number 2 is that if we find these cell type specific context dependent QTLs, and they are at least colocalizing with the disease, it's already a huge success, because usually we find like hell amount of the eQTLs, hell amount of jivas variants. Nobody knows what the hell does it mean. If there is a match, no, well, people know. Maybe I don't know. Okay, so my supervisor will see it and is like, no, so will not approve it. But idea is that there is can be crap on one side, can be noise and issues that we will talk afterwards on the other side, and it's already a success if something that you find from two perspectives colocalize. So with this, our group, my research group in Ludwig Franke Lab in Groningen, we initiated an amazing consortium where more than 2,000 of samples, single-cell samples, together with genomics were analyzed. And here we studied the single-cell eQTL effects in blood. And here you can see that our goal is to understand what is the eQTL effect in one cell type, what are the cell type specific EQTL, so the same QTL effect not present here, but also what are the stimuli dependent effects, as well as how much it all can be built to the network and like put some hints into the disease mechanism or preventive medicine interventions. We are not alone, but even though having this success, having thousands of associations, we still face many challenges. Challenge number one, when you inherit genetic variants, it's not like one-to-one inheritance. You inherit this into the blocks due to crossing over, due to the linkage disequilibrium, like szczeplone наследование, then you do not inherit a single variant. It implies that if you find the signal from one SNP, then the signal from another SNP, it might be still the same signal because these SNPs are inherited together. It screws out your false discovery rate or like other FDR procedures. But the idea that you need to be aware that even though what you find might be very much impacted by the linkage disequilibrium and enables you to find the causal variants, like exactly the variants that make the action. And also, if you find the associations, like if you do only EQTL analysis, we are not that much further from JIVA studies. We still don't know the mechanism. We know the associations. This is the genetic variance. This is the gene level which is affected, whatever. How it is affected, what are the transcription factor binding, what are the regulators, we don't know. Meaning that there is a no network approach as soon as you only do the EQTL analysis. So now I will suggest, I will present two different parts. How can we work? How can we live with that? First of all, if you work with single cell data, you can utilize the benefits of Multiome, and in case you find several EQTLs, imagine like 3 different SNPs are affecting the level or like are associated with the level of expression of one genes. So first, if you know if these SNPs are located in the open chromatin region, you can already kind of prioritize that like this SNP number 2 is less likely to be causal because we expect that the causal SNP is inside the open chromatin, because if something is used, it needs to be open, it needs enabling the enzyme to attract it and to attach it. Also, what we can know, understand is that how much the activity of this open chromatin region, again, we talk about multi-omic usage, can be used for the understanding this gene regulation. And if we see that this region is correlating to the level of, like openness of this region is correlating to the expression level of gene, and this is not, then this region and this SNP is likely to be causal. This is kind of a very, bold analysis, like correlations or overlap, bad tools, like not a rocket science, but still, it's important to do this properly. So lastly, you can find, zoom into this region, and then find out the potential regulators, meaning that if this SNP is located within the open chromatin region where the particular transcription factor is binding, then there is a very likely high chances that this SNP is causal to affect this gene, and maybe causal to be affecting the whole phenotype from the JIVA's perspective. So, to close the part number 2, I will suggest 3 main strategies. How can we not only assume or narrow down the signal, but also prove the causality of the genetic variants which cause QTL effects? In our case, expression quantitative trait loci. First, we can check if in silico methods this genetic variance is actually disrupting the transcription factor and binding site. So we know that, for gene to be expressed, there needs to be open, like the helicase need to work, the DNA has to become open, but also the regulator or transcription site needs to bind to initiate the transcription. Meaning, transcription factors often do not bind randomly. There are some that do, but with a high regulatory potential, they don't. They have a particular palindromic repeats, recognize that the molecule recognizes itself and then knows that that's the place where I bind. But these palindromic repeats are just oligonucleotides, like the sequencing. And if the genetic variants, or single nucleotide letter, is changing from one to another, the affinity and the binding of this transcription factor can decrease. And if we observe this, we can state about the disruption of the transcription factor and binding site. So for example, you identify this SNP as an EQTL effect for FOXA1 gene, for example. But also you find out that this, it's not a gene. Okay, you identify this SNP as being the EQTL effect causing SNP. And then you can see that this SNP is also making the part of the two transcription factors, disrupting these transcription factors in the same position, meaning that there might be that if this SNP is disrupting this transcription factor on binding site, and it was previously reported that this transcription factor this transcription factor is regulating this target gene, there might be a link that actually this SNP is likely to be causal, and this transcription factor is likely to be causal in terms of regulation. So basically, transcription factor and binding site disruption can be done with motif breaker in our relatively straightforward tool, which allows you to benefit from the data sources collected from 10 years till now, and then assume and check out how your genetic variants behave. Number 2, we can check if the variants we're working have any of the function. What does it mean? Majority of the genome is not used. It's not a new information from you. Majority of genome is silent, is not active, is kind of junk genome or whatever. And not only all the variants, which can be even found as an association, can have this functioning. To understand if the function, if the genome region and the variant itself has any functioning, we can conduct, again, like the CRISPR-I screens, like coming back from Andrew Bass's lecture, or MPRA, massively parallel reported sequencing arrays, where you test the functionality of this variant. So basically, you create the set of synthetic promoters, like other nucleotide sequencing, to further incorporate in biological system, and do the readout to check, okay, the synthetic promoter number 1, did it change within my biological system expression of the gene, or like target genes, or all genes? If not, meaning that this kind of change or genetic variation doesn't make any sense, like it's not that functional. If it doesn't cause this within the biological system of interest, it can be, of course, like not the well representation of the biological system, like because it's still in vitro, not in vivo, but in majority of cases, it says us about the absence of function of these variants. And the same you can test, like if another oligonucleotide is giving you this hint, then you can say, okay, this is the region where I'm more likely to find my causal variants. So, and now imagine this kind of second block converted and like become on steroids. And now imagine like to learn the kind of insights from this massively parallel, MPRA assays and CRISPR-I screens to the network and the machine learning model, where you can predict the functionality. You don't need to redo all of this experience. They are super time consuming, absolutely like deadly impossible to reproduce in a lab, but still possible, but to some extent. So basically you can now use the machine learning tools to understand whether your genetic variant is actually having any function. This was done by Former, this was done by Borzoi model, recently published Promoter AI, I think from Google. But the idea is that now you can even have your variant and then test how much this variant is associated to the changes from the histone level, DNA, Steve binding, in expression itself. Basically, these 3 components give you the information. Okay, do I look into the right variant? How do I tackle the issue? Like if you have the LD variants, like hundreds of variants, and then test one by one, and then you have like this course, okay, this variant is disrupts motif breaker, by motif breaker, this variant is predicted to have a higher functionality, then you have more hint to narrow down the list from hundreds to tens, or even a few numbers of these likely causal genetic variants to further test in a wet lab. So, recap, and coming from the most spicy and juicy section of this talk, is that, like, recap from part number 2 is that quantitative trait loci analysis is a very classical statistical procedure, invented like 10 years ago, but still very informative to understand the genetic signal from GWAS traits. Having at least put in the light of how genetics is, well, it is a light, but how genetics is affecting the molecular layer of information, and further can be linked to phenotype. To recap those colloq analysis, MPRA screens, and the EQTL analysis with overlaps themselves. So basically, with this, very important to know this, to make this properly, you can work with a single cell RNA-seq data set, single cell multiome, but you do need to have genetic information. Alternatively, there is like a little specific expression, but in the ideal scenario, you need to have your at least genotype donors to make these associations. And in the end, you can have a very nice discovery to interpret some of the disease mechanism and like make a step out from fundamental science to something more translatable. And then let's, for the last 15 minutes, let's dream together and talk about the future perspectives. I prepared 8 different future perspectives that I personally find the most fascinating and deserve the attention and talk. So first of all, what's yet to come? Even though there are more than 100 different single-cell protocols developed at this date, there are even more to come. More developed in terms of the analysis of the different modalities. So that in terms of the enhancing signal throughput of the cells, meaning that, again, having these kind of combined methods and either single methods that I totally didn't cover in my talk, having this information about this, like the comprehensive view of the cells captured by different 3-model, whatever, N-model protocols, we can now work and understand with this biological system much better. Also, from the protein and metabolite perspective, these single-cell technologies, they still exist. They're just not that much NGS-based. They still exist, and the focus is to isolate your cells and then treat a cell volume as a small kind of aliquot or like whatever this vial for the further HPLC-MS analysis and so on and so forth. But this type of protocols exist. This is scope 2 protocol. Unfortunately, I haven't put this on the slide, but so far you have to trust me. This analysis exists. But even within one modalities, without the need to come to other whatever research methodologies and techniques, we still have so much discovered and unstudied in terms of at least the level of RNA, unspliced RNAs, like the mechanism of splicing themselves, the non-coding RNAs, the post-transcriptional RNA modifications, or like the processing that are in general happening to regulate the RNA activity, ribosomal occupancy, like the binding and like the transition of the matrix RNA within the cellular space. It is all unknown and needs the advanced technologies. They are actively developing. Phyllis does not state on the how it states. But still, we need to be ready, and we need to know that even though it sounds super complex and interesting, there is so many to discover else. So, point number 2 is that, as I said, in a human cell atlas, there are more than millions of cells, hundreds of millions of cells. And it's not the only thing. It's like it's still coming. This research is ongoing. This kind of technologies become cheaper and cheaper, and like this scale of technologies is increasing. And what does it mean for you? For me, it means that with this race of data, there is also the freedom to conduct the bioinformatics and purely data-driven experiments without the need to stay in the lab for years and isolate your amazing PBMCs. But ideally, it's kind of the glory and the wealth of use this data as this data is coming from single-cell RNA-seq, ATAC-seq, multi-omics, and so on and so forth perspective. So for some of you, that might be a luxury to contribute from the data generation side, but also everyone who will be starting with single-cell can contribute from the data inside generation side, like from this data processing. As the data is here, we don't need to reinvent the wheel. Point number 3, spatial context. My talk hasn't covered at all the spatial context, meaning that, and there could be one reason, I've been working with blood. Blood doesn't have the spatial organizations. It's a liquid form, like spatial doesn't matter. But most of the tissues, like brain, liver, and so on and so forth, they are organized in a 3D and space, right? So to properly grasp it, you need to apply the single-cell technologies. So from organ, don't go for bulk, just homogenize it all and then see how many of, like, what is the average color of these Legos. Don't go for, like, okay, go for RNA-seq, but if you have resources, go for spatial RNA-seq, where you are able not only understand the cell type proportions and abundance, but also the spatial organization of your sample. Especially it's relevant for transplantology, for diseases affecting this kind of inflammation flow, signal transition, immunology, and so on and so forth. What is, but what is also more important, that I just showed you 2D model with the advance of the with this advanced algorithms. You can even recapitulate 3D model of your sample, having this properly organized sliced, and the software to integrate it into one signal. I'm super proud because unfortunately I didn't stay in the lab till that moment, but this protocol, OpenCT, was developed where I've been doing my master's. So it's super exciting. If you've been working with single cell spatial data, definitely check this out. It's kind of the mainstream. It's, from my personal view, is one of the most prospective directions. So, circling back from perturbations and level of single data, we talked about the genomic perturbation from this Ampere array perspective, but what is the perturbation? It's kind of any kind of change that you can introduce to your cell and biological system to understand what's going on. The thing is that it is always better and always more interesting to study it in comparison. Like, this is cells that haven't seen the stimuli, this is cells that have seen stimuli, like this is cells from vaccinated person, like this is not, and so on and so forth. So this kind of stimulations or like genetic perturbations, which might be relevant for this case, like these cells are with this transcript, these are not. This is kind of analysis that gives you the predictability to assume what can be changes to the further like similar molecules or similar actors. Meaning that, again, sorry, I thought I had the slide. So, meaning again that you can also predict what will be happening within your biological system if you have the knowledge of the previous, like, similar stimuli. Almost there. So, in the middle of the part 3, I would like to point something that is not necessarily as a pain or perspective of the single cell, but as a populational genomics and human genetics, where I also consider this my major field, that the problem is that in the majority of studies, this is kind of the distribution from GNOMED, in the majority of studies, there is a non-equal representation of samples. Again, we mostly work with European samples, 55 plus, mostly females, because this is kind of the people that are more into being a volunteer. I don't know, maybe every female will understand, but they're like more proactively to be involved in the scientific research on the voluntarily basis, or and so on and so forth. So, and it's only about talking about the background and genetics, meaning that there are many kind of stimuli, many kind of like metadata points, like I treat it in metadata in my whatever S3 object in my head, but there are many type of interesting points that are not necessarily covered from the point of genetic, from the point of single cell research, or in general in-depth observation. It means that with the rise of biobanks, with the democratizing and dropping the price for sequencing technologies, we will have much better situation there. I truly pray for that. So, this is something, I don't know, have everyone seen, okay, have everyone heard anything about LifeSeq before? Okay, I'm super proud. So LifeSeq, I'm super proud of myself that I have this kind of luxury to educate, because when I've read about this method, I think 4 years ago, I thought it's a revolution. It's like literally something that will change the world. Idea of the LifeSeq is that, okay, idea of single cell is that to study yourself, you need to fix it, you need to isolate it. Okay, in the end, yourself is after a lysine buffer is dead or like dissolved. Okay, that's the word. But imagine if you can study yourself in a non-destructive manner. Okay, it doesn't sound like that easy, and of course it's not something I have this kind of in my lab. It's super expensive, but the idea is that you immobilize yourself in a particular syringe, or like the thing that is able to aliquot the sample volume time to time. And then you can study like this micro volume, like picovolumes of the cells from this LifeSeq protocol from Bart Planck lab. Amazing work. So you can study what are the changes of your mRNA level without distracting the cell, or like without not only mRNA but from biomolecule perspective, like what's going on. It's super exciting that it's revolution. And in case if it will be put it on the scale, I believe that it's like the next new black, this new revolution will happen. But of course, it's difficult to scale, because imagine how many syringes you need to have for 3 trillions of cells in a human body, or at least one research experiment. So, circling back that we know, I by intent have not put this kind of Moore law about like dropping sequencing price, but we all know that the sequencing prices are dropping sequencing itself becomes much more accessible. Like the 3D genome sequencing is coming with like promising like up to 10 euros per a whole genome 3x coverage looking forward. But the idea is that everything is democratizing. The scaling is easier, then you can easier generate your sample, generate your libraries, and so on and so forth. So everything is going good, meaning that for you in future, if your lab will be considering doing this method, it's non-zero probability. So it's like pretty much likely. So it's, again, lower cost, more data, better insights. And lastly, my favorite is the foundational models and systems medicine. So when ChatGPT was released, I think after half a year, I've been to a conference, UMCG last year, where Fabian Theis presented the cell GPT, or just touch upon that. I read that single cell GPT is the foundational model which allows you to understand basically the changes and dependencies across modalities, across all the data sets which are happening. So it's kind of operating in a similar manner. Don't ask me how, I don't know, but it sounds fascinating. So if you are into LLMs, biology, and like data science, like deep tech research, consider this. I would state the foundational model in general, narrow it to a particular disease cases for like whatever digital twins or like in silico drug research is super exciting. It's like amazing. And with this, it's kind of my personal future perspectives, like which are covering, again, like all three parts that we know that this kind of single cell is super amazing and like extremely informative. We know that the single cell is moreover useful for understanding and stating the genetics question. And this field is not just like appeared in like one day splash. It's a very, very rapidly developing field. It is amazing. I truly invite you to discover it and find your place there. And I hope that this was, my talk was at least a small hint for you to raise your interest to zoom into the specific parts which are relevant for exactly your scientific project. Because in any lecture, it's not possible to cover something widely applicable. So with this, I would ask you to, for my last minute, to raise your hand. How many of you understood and kind of have a little bit of kind of this improvement of understanding how can we use single cell after this talk? Okay, so fair. Thank you. It was not for nothing then. Okay, so I'm happy that we all have this kind of tool set to have this better understanding, and I really appreciate your being active and like online audience. It's not for you, but I hope I will have questions. No jokes, but I really like that we discuss these actions today, that we covered again single cell, the EQTLs, and like Jiva's story, and a little bit of future perspectives. And for those who will contact me and who would like to have the slides, I also have a little bit of a bonus, which is like the little bit of pipeline. You can go there now, but this is kind of the pipeline, how I see the processing and the selection of the single cell methodology for this. I know we have no time, so in brief, I will just scroll how many of these, like what you have to consider, like this is all in the QR code there. Share it, whatever, do what you like. I, how to say it, recommend this tool, Eraser AI or IO or something like that. So in brief, there is something there, and many thanks for you, for your stamina, for managing, listening in this talk. That's it, thank you.
Okay. Thank you very much, Maryna. And now it's time for questions. You can use both Ukrainian or English language as you wish.
Just raise your hand.
Це не страшно.
Please, you can raise your hand and ask Maryna.
Yeah, please.
Oh.
Hi. As I understood, when you want to isolate one cell, you use this droplets method. When you, I suppose, dilute the cells to the point where this one droplet, one cell. Yes?
If I understood your question properly, first of all, you're never interested in isolating one cell. You would like have millions, but I got your question. It's a small
Yeah,
joke.
so my question
if
is
you dilute to get the
one cell in one droplet, yes?
Yes, your goal is to that one cell is ending up into one emulsified droplet together with a magnetic bead, which is this.
So how can you control that there is
One cell.
one cell, not
Yeah.
two cells with this magnetic bead and
Yeah. Or like 0 cells in this, like
or zero cell, or maybe some extracellular RNA
Yeah.
with this marker?
Yeah, so it's a very right question. And if you haven't been working with single cell, it's a very proper question to ask. Reality is the follows. You have these crappy data points in the end, like doublets, ambient RNA, 0 states, and so on. And how you control it on the level of this kind of encapsulation of the cells is that like you use the proper dilutions and control the stream. So indeed, your goal is to have this kind of suspension of the cells from one solution, which will follow make the emulsion of it together with bits, but knowing the concentration like this kind of volume in one suspension, you can further estimate and kind of catch how much of the cells are ending up into the final library. Reality is very different. It's like 10% of cells are luckily if any cell is getting into this violet or like into this droplet. It's not like one-to-one. If you would engineer it by hand, it would take years, but maybe you will get it. But high throughput methods are working on this probability, so you kind of have this volume, 20% are gone, like 10% are remained, and then you end up what you end up with. But yeah.
Any questions more?
Okay, 2 questions there.
For the lecture, I have 2 questions. First of all, are single cell methods are used for diagnostic or not, or it's no need?
So there is a specific cycle that something becomes a diagnostics, and single cell so far is something for fundamental research mostly. However, my expertise for translational science is very narrow, so I assume there is something maybe coming from the CAR T-cell therapy, cancer therapy, and so on and so forth. I assume that something might be there for diagnostic. I am not the expert in that. Healthy blood is my stream, so ask me about healthy blood, so yeah.
Thank you. And the second will be about LifeSec, because I took part in bioinformatics school, and there was a question about LifeSec in human brain. Yeah.
Okay.
But and we have discussion about that. But LifeSec is something closer to that. And could you explain more how it's conducted, and can it be conducted maybe at the organoid level?
Yeah. I don't have my slides. Ideally, you have the same single cell setup and the emulsion of the cells. And again, I will explain not how it works, but how I understand it works. So check the Bart Planke, the Planke lab. LifeSec, proper preprint is there. Idea is that you take your cell, you immobilize it with like to the specific syringe, like pipette. So this cell is kind of attached, like similarly what you do in echo, like and so on and so forth. And then you put this within this like attachment of the pipette. You put the syringe that then make the canal to extract some of the volume of the cell into the volume what you would like to. And therefore you analyze this volume, because here it's not like the throughput. You don't analyze tons of cells. You analyze 3, no, like I don't know how many, but a few. So ideally that works as such. You have like a living cells attached to something with the syringe, like with the canal that is further extracting the biomolecules in a time-resolved manner, for example.
Mostly it is used for like separated cells, yeah?
Yeah. I cannot imagine it works in brain. I cannot. It's not a. I can imagine for brain organoids. I've been working with them. These guys are like put some enzymes, like they remove the cell connections, matures. How say it? Like matured neurons are not that whatever, not that stable. But like I cannot imagine for a in vivo brain. Let's call it this way.
Thank you.
More question?
Okay, there is here, but who?
Thank you a lot for your lecture. I wanted to ask, so as I understand, because of the single cell sequencing, it has a lot more data and a lot of dimensions in the data. Do you have problems with the additional noise, and how do you deal with that?
Yeah, so indeed, single cell data is extremely sparse and noisy at the same time. And importantly, that this level of sparsity and noise is specific for a modality you work with. So it's uncomparable to match attack and single cell. So if you talk about the RNA level, there are many well-defined techniques on how to reduce noise, how to do QC, and how to maximize the signal in the end. In the reality, you're totally right, it's multi-dimensional space. You start with like 20,000 of genes, 30,000 of genes, like 60,000 of genes in terms of SmartSig2. And your goal is to end up with those genes which signal you are confident enough, because it's unhandable to work like hundreds of thousands of cells with like tens of these genes and so on and so forth. So basically, after doing all of this QC, prefiltering signal, prefiltering cells, you find these mostly variable genes, which are mostly often like the cell type markers and so on and so forth, but genes which are kind of representing enough your genetic programs. And based on that, you kind of state that, okay, I work with single cell data, and I knew that I captured 30,000 of genes for like 20,000 of protein coding, but reality that I work with 5,000. And then it's like the signal space that you are looking at. But it is noisy, it is sparse, but there are tools to handle this, yeah. I was...
Hello?
Oh, okay.
Yeah, was a nice lecture. I barely understood like how the biology works, but I want to ask like what opportunities did the UMCG lab at Groningen give you that wasn't anywhere else?
Okay. Well, I just got it about UMCG and I got heart attack. What was the question? Sorry, can you repeat your question? What I did in
What opportunities did the UMCG lab give you?
Gave to me?
Yeah.
Well, we're on stream, so let's talk later about it. But in general, it's amazing environment. Lude Frank is a fantastic scientist, like in the top 5 in the field of functional genomics, like very high impact scientist. And I am under supervision of 2 amazing supervisors of Monique van der Wijs and Mark-Jan Bodder. They're superstars. It's like literally, they guide me. They gave me the way I can be self-critical and tackle the hypothesis. They kind of cutted my wings as a young student, stating that like, okay, I can do every type of multiomics, and I was like, we do single cell for this type of population, and that we publish. So, like literally, it's kind of the school of life. But they are amazing. They gave me many opportunities to study, because this is like a very computationally heavy lab, not a wet lab. I was one of the few wet lab scientists, but yeah, opportunities to study. Find your supervisor, be critical and be selective for your supervisor. Is the person who's kind of more important than husband? Like, well, okay, okay, I'm also married, but in terms that is like, okay, you got my point.
All right, so there are more opportunities for someone who's not like wet lab scientist, more like computational, like statistician, more specialization. Would you recommend someone to attend that?
In that lab, yeah, for sure. There are some of the computational, like UMCG is famous for computational research. We have this center of the human aging, like the microbiome group, amazing. So like Netherlands is in general one of the top countries for me to do science, like together with Germany, Switzerland. And so consider this heavily, like, but be specific of your research interests. If you don't have a passion to EQTLs, don't approach them, but yeah. I didn't have passion either, so I'm just saying that.
Yeah.
Yeah, so questions? Yeah?
Thank you, Maryna, for such astonishing lecture. Back to the modalities.
Yeah.
And can I ask you, can you speculate or maybe say something about the missing modalities, like what we start to say maybe about metabolomics, maybe lipidomics, and even less?
Yeah.
Some molecules which not. Can you say something about it?
So indeed, I didn't elaborate that much on the future perspective, but about like extending to modalities is very promising from my point of view. So the whole idea is that we kind of can do really a lot from NGS-based methods with like full-length RNA, like together with DNA itself or complete number of variations, so on, like open chromatin and all the DNA modifications, because it's something that ends up in like whatever shotgun library. So it's something that you can see from the sequencing like in your FASTQ files. But from proteome, like protein sequencing, protein modification, protein abundances, like metabolite modifications, metabolite presence and other kind of modifications together with lipidome, it's all about HPLC and gas chromatography or gas chromatography or the mass spectrometry and so on and so forth. So the whole idea of this type of analysis is that you tackle it from other way. Usually, this kind of workflows, like it's from DB Rhapsody, something I've seen, something like that. Your goal is to isolate the cell into one single volume on a nanowell, and then extract one by one this kind of the pool of molecules based on the physical properties that you are interested in, and then kind of enrich it and come and go for mass spec. But like again, pico volumes of cells, like this even smaller volumes of this few metabolites, and so on and so forth. But I remember that 5 years ago when I was giving a lecture of single cell in Bohumolets Institute, it already was a preprint about the spatial lipidomics and like spatial single cell lipidomics, where you can properly tackle the isolation of this protein, of this lipids, and then kind of be able to decompose from this content perspective. So if you Google single cell something, you will definitely find it. But my kind of guess is that there is just a huge distinction that the metabolites you cannot tackle in this metabolite-wide manner with NGS methods. So that's kind of the disadvantage of that, but also a luxury that at least genes are covered. So, yeah. So,
whether we have additional questions?
Okay.
Yeah.
Let's make it the two last, yeah, and then. Yeah, please.
Я скористаюсь можливістю задавати питання українською.
Окей.
Ви вказували великі мовні моделі і казали, що вони можуть бути корисні. Я бачу безліч напрямків, а от якщо б у вас була можливість в чата GPT запитати конкретне запитання, то який саме напрямок, тобто як би ви скористались великою мовною моделлю для того, щоб отримати інформацію? Тобто, що б ви в неї запитали?
Я українською можу, да?
Так?
Да. Якби я створила свою foundational models with single cell, я би продала це Big Pharma як digital twin, для того, щоб робити research. Я вже над цим працювала трошки, просто я вирішила трошки на паузу поставити. Але, да. Ну, ідея в тому, що ці великі мовні моделі можуть зробити багато in silico perturbation від філоаналізу, тому це дуже потужна річ. Це або drug repurposing, або research, або marker discovery, тобто це більше, або, ну, все-таки фарма для мене, це більш комерціалізуємо в мою сторону. Да? Ні, питання щодо довіри до даних AI, тут вже ми говоримо не про AI, який там хтось там Gemini, OpenAI викатив, ти саме ти створив, як своїм там контрольоване середовище з твоїм інпутом, аутпутом. Ти його валідуєш або на рівні даних, ну, типу, бачиться те, що ти очікуєш, або ні, або вже, якщо ти всьому віриш, то вже в мокрій лабі. Тобто це не silver bullet, яка тобі, ну, без мокрої лаби ніхто не виживе. Да, мокрі сайентисти, ми тут, але тим не менше, да, хоча б, щоб мокрі біологи не перероблювали все одразу, це хоча б, ну, визначення напрямку, куди ми можемо рухатися. Я думаю, останнє питання, якщо буде.
Yeah, we also have some questions from the online audience. So the question is, could you comment whether it's possible to determine the chromatin entropy using single-cell sequencing methods and use this data as an agent markers?
So first of all, I'm not familiar with the term chromatin entropy. I'm familiar with chromatin openness, and then of course you can identify the chromatin openness with the single-cell methods. As such, single-cell multiome, single-cell attack, and so on and so forth. But as a marker of aging, I'm hesitant because, to my knowledge, aging is more linked to the methylation profile, not necessarily. Maybe it was your question, basically. And I am not the aging specialist, so I cannot comment on that. So I know that there is a lot of this Hayflick scales and so on and so forth, and probably you can do so. But as far as I know, you do this in blood, in bulk method, and then do the traction. So it's not necessarily something that single cell is applied for. But I can assume that this aging is happening in the different tissues in a different pace. So no idea. That's the honest answer. Okay.
Yeah. Okay, whether we have another questions from audience? Yeah. Please.
Oh, okay, so only two. Yeah, go. Oh, it's like the lady.
Okay,
Yeah.
I have a very fast question. What is the bare minimal cost of single-cell transcriptomics, assuming that you have all equipment?
Oh, then what are the costs you would like to know? The reagents.
Yeah.
I cannot. I don't know this information. I know the ranges that can be from 25 to 1,000 for one well. So it's like it's hell expensive. When I was saying about hundreds of thousands and millions, it was not a joke. No, not overestimation. Yeah. So it's super expensive. That's why I'm like, data is generated. Like, go ahead and use it. Not, yeah.
And you did 2,000 of samples.
200. And my consortia did 2,000, but it's like I'm on several labs. But in my lab, we generated in total 500, and in my research, 300 samples. Yeah, and once one lane had failed. It was a heart attack for everyone in the lab, so I cannot tell you. So, yeah.
And what next generation sequences platform do you prefer?
Well, I am on the data analysis side, so I don't dictate my rules there. So no comment. Like it sounds like your question sounds provocative. Like what are the commercial provider I would recommend? I'm not providing. I'm not paid by that. So no, sorry. Yeah, but well, I use only shotgun sequencing. So then I approximate. So we can talk afterwards if you're interested. So also, and the last question.
Okay, it will be quite small question. How the first sample, I mean, how the clarity of the sample at the beginning, I mean, like the blood or brain cells impact on the quality of the single cell data. In my mind, if I understand it correctly, it's easier to get the proper database of single cells from, for example, blood than from muscles or other more complex cells. Is it like correct way of thinking, or it's not so big impact, or if it is, if there is some methods to manage it and to work in single cells, not only with blood, but with other cells, and if it have some perspectives for future?
Yeah, absolutely. So absolutely the sample accessibility is the key. Blood is the easiest because we all withdraw blood for donor or whatever, lab diagnostic procedures, and none of us have withdrawn whatever brain, right, or a muscles biopsy. So it's like, it's very painful. And majority of the samples from complex and non-dilute tissues are got post-mortem. And that is why there is a huge imbalance from blood. So blood is easier to get. So with our consortium, we onboarded these 2,000 of samples together with genetics. With a similar consortium, when we are tackling and studying the same cell type specific effects in brain or gut, we are generating, like we were able to analyze like hundreds, not thousands, but hundreds of samples. And of course, it's all about the sample accessibility. Like gut biopsy is not that common procedures like checking your hemoglobin level where the blood comes. So the progress is there. So again, I assume that things will not change. Complex tissues will come from the post-mortem samples in most of cases, or some biopsies like gut for Crohn's disease, muscles for myopia and so on, myotrophy and so on. But yeah, it's difficult to change. Even another liquid tissue like liquor is not that easy to extract due to the pain. So blood is a luxury to work with. If my experiment was failing, I was using my blood to test. So that's kind of okay. Which ethics is not approving, but yeah. So I always forget that we're on stream, and then I'm so open. I was like, Jesus Christ. Okay.
Okay, it was last questions, and you can ask more questions to Maryna in informal communication afterwards, after lecture. So let's thank you, Maryna. Thank you. And thanks to everybody who participated in such an engaging discussion. We really thank you to join us today, and hope you will you have had a great time here, and you can then continue to do it, talking with all friends and maybe finding potential partners during our networking sessions, which will start soon. Coffee and snacks are waiting for you.
Yeah, and to wrap up, we actually want to make a little bit of announcement. So please, next slide. Yeah, so our next lecture in the BioGENext series will be held on the 23rd of October, and this lecture will be hosted by Boris Olefirov. He is a researcher at the Department of Biophysics of Sensor Signaling at Bogomolets Institute. And he will have a lecture on the advanced microscopy and image analysis. Actually, it's a little, in some way, complementary to what Maryna already said, because she already mentioned that it's quite important to employ proper image analysis techniques to cover the special transcriptomics and special omics technologies later on. So Boris will cover a broad introduction into the advanced imaging, into multiple modes of imaging that is used in current research, and on the tool set and toolbox that is utilized to efficiently process and drive some biological insights and biological answers from that imaging data. So you're really welcome, and this lecture will be held, will be hosted by National University of Kyiv-Mohyla Academy. So please stay tuned to our social networks, to our announcements, and be welcome to be part of this future lecture that will be held by Boris. And another announcement is actually after the lecture, during the networking session, we will have here the area, lighted area with a wall where you can take some pictures with your friends, with others who participated in this event, to memorize for yourselves this wonderful time to learn about single-cell sequencing together. So thank you all. Thank you very much for joining us together. And let's start and continue our BioGENext series season 2. So nice to see all of you, and have a nice time during network session.